Okay, great. So thank you, Jenny. As she said, I'm a graduate student in Dr. Jonathan Berg's lab, and I've been working with the FBI to help develop some recommendations for applying the functional evidence criteria PS3 and BS3 for clinical variant interpretation. And so first, I want to, for anyone that's not as familiar with the day-to-day codes that people talk about, I'll be talking about the PS3 and BS3 criteria, which say that they are well-established functional studies showing either a deleterious effect or no deleterious effect on the gene or gene product. And this counts as strong evidence in the Richards et al. Framework, which also provides some considerations regarding functional evidence, but doesn't give a lot of detailed guidance on how to evaluate functional assays for variant interpretation. And differences in application of these codes represent a major contributor to variant interpretation. And I will also say that for anyone that was on the ClinGen on Friday, I'm mostly presenting some of the similar information with a couple of added details, and I hope we'll have some additional time for discussion, as I know that you all are often those that are actually applying these rules. So before I talk about the recommendations we've made, I'd like to first acknowledge some of the work that's been done by our group at UNC to address this issue. And some of this work has laid the groundwork for the recommendations that I'll present later. So first, what we asked was, what defines a well-established functional assay that is suitable for the BS3 or PS3 criteria's application? And then what data or metadata is needed to make that kind of determination? And how should this functional evidence be structured for curation and for computation, so putting it into the BCI? And our approach was to critically evaluate the 6 BSEP gene disease rule specifications that have been published as of April 2019 in any primary literature that they've cited. We examined rule specifications, BSEP approved assays, and assays that were considered but ultimately not approved for PS3 and BS3 use. So while there's lots of data that I could talk about, because this was a tremendous amount of work, primarily led by 2 of our postdocs, Shannon and Donna, as well as another person in our lab, Mira. This is kind of the overarching summary of what they found, that there are 4 different parameters: replication, controls, thresholds, and validation measures that were identified by at least one group as being important for their PS3 or BS3 guideline specifications. And we evaluated how often they were specified by each of these different BSEPs and whether they provided any recommendations for modifying the level of evidence to assign a PS3 or BS3 classification. So either keeping it at this strong level of support or downgrading that to a moderate or supporting level. And so you can see that there is some difference in what the different groups consider to be important and even what level of strength they assigned those evidence codes. And overall, I want to share some of the big conclusions of this analysis that were useful in developing the recommendations of the SBI. So we found that VSEPs varied significantly in how they defined which assays were well established and which parameters were considered important, and ultimately how they applied those specifications. In particular, more guidance is needed on how to estimate the predictive power of functional assays, the consideration for how to use splicing assays using the PS3 and BS3 criteria, as some groups applied splicing criteria under the PS3 or BS3 code and others did not. How should we be using patient-derived experimental material in functional assays, whether there are any specific recommendations for using model organism evidence, and any guidance for conflicting evidence, often either different laboratories performing the same type of assay or 2 different assays may give conflicting results. So many of these points were then incorporated into the recommendations that we made with the SBI for the PS3 and BS3 criteria. So this is a group that's helped develop these recommendations that we have since submitted for publication. So first, to look at the Richards et al. Guidance, we decided to elaborate and clarify some of the points that were made regarding functional evidence. Important considerations for functional assay validity included the biological environment, so what materials are being used, is it patient tissue, model organisms, or something else in vitro, and the genomic context, so which functions are being assessed, as well as reproducibility and robustness, which they state is mitigated in a CLIA laboratory, invoking that the entity performing the assay is important. And finally, they suggest that mRNA and splicing assays can give important functional data. So we expanded upon these different points with some specific recommendations. First is the issue of patient-derived material. When using patient-derived samples, a functional assay actually is evaluating a broader genetic and physiologic background, so the other variants that are in cis or trans that might have epigenetic effects or that may be specific to a cell type rather than just the one variant in isolation. So it's generally better to use this evidence to satisfy the PP4 criteria, which refers to a specific phenotype, and then to delineate the expected phenotype in patients with certain combinations of variants or homozygous variants of known pathogenicity. If the VSEP then decides that they want to use an assay performed in patient-developed material, then they should apply the criteria at the strength level determined by the validation parameters, not simply based on this being in patient-derived material. And I'll get into what validation parameters we're referring to here in a little bit. They should also specify if there's a particular number of unrelated individuals in whom they think that the variant needs to have been tested in order to qualify for evidence and variant interpretation, to help mitigate some of those issues of the broader genetic and physiologic background being assessed. For model organisms, for clinical variant interpretation, using functional evidence from model organisms should take a nuanced approach. So considering how well does the model organism reflect the human anatomy, physiology, genetic context, measurable phenotypes can vary. So does the model system display a comparable phenotype? Compensatory background variation can also affect the phenotype. So are there any other strains or lines that were considered to mitigate those effects? And if you do use model organism data in variant interpretation, the strength of evidence, again, should be adjusted based on the rigor and reproducibility of the overall data, not based on the model organism itself. This is genomic context. So that refers to the nature in which the variant is used. For example, you could consider potential off-target effects of CRISPR-introduced variants and the limitations of cDNA expression, which can't assess splicing effects. So a result supporting a normal protein function, for example, might not capture any abnormal splicing, and so maybe benign evidence should be assigned a lower strength in light of the limitation. So while it is preferable to test variants.
There's some real background noise. Is that coming from you, Sarah, or is someone else needs to go on mute?
I think that I am hearing that also. I don't think that that's coming from me, but I could.
Can everyone just check their mute and make sure that you're on mute? Thanks.
Thank you.
Well, I don't think it's coming from me.
Okay, I think we're good now. All right, so while we believe that it is obviously preferable to test in a more natural genomic context, it's not a requirement of a well-validated assay. And when assigning evidence strength, you should consider how the approach impacts the interpretation of the results and how the study controls for those limitations. Next is splicing. There's no doubt that RNA splicing assays provide valuable information in clinical variant interpretation, but there's been some questions about whether this criteria is the most suitable for applying this evidence, or whether there's another criteria that would be better suited for this type of evidence, since RNA splicing doesn't provide a direct measure of protein function. So for the canonical +1 and 2 splice variants, the splicing assay results can actually help refine the application or enhance the strength of the PBS1 loss of function criteria using the decision tree as recently laid out by the SVI, currently the predicted impact of the variant on mRNA stability and protein reading frames used in that assessment, but you could then add the splicing assay results to help upgrade that strength. Also, for variants outside the canonical splice sites, there is an SVI splicing subgroup that has recently formed that will start to address how to apply evidence of predicted altered splicing. For variants in which the splicing assay shows an impact that's not predicted to lead to loss of function, that might be in-frame exon skipping or a large in-frame deletion, they may still be able to apply a PS3 or BS3 criteria if the assay looks at the protein function of the resulting product. And so that second assay would be more appropriate for the PS3 or BS3. Next, we recommend that groups describe functional impact of a variant as measured in a given assay as either functionally normal or functionally abnormal, and then giving more specific granular specifications as outlined recently by an Enigma group, where the functionally abnormal could be a complete loss of function, a partial loss of function, or intermediate functional effect or hypomorph, gain of function or neomorphic, or dominant negative. And we specifically chose these terms, functionally normal and functionally abnormal, because we've seen lots of different terms thrown around in the literature describing results as benign or pathogenic, but that really falsely equates functional impact with an interpretation that involves actually a number of other evidence lines, not, and it would be inappropriate to look at an assay result in isolation and determine that the variant is benign or pathogenic. We also recommend you not use the words deleterious or damaging to describe the result of an assay, since that typically only applies when a loss of function is the mechanism of disease, and not gain of function variants, for example, which could lead to disease but may not be damaging to the protein itself. So the final assessment of the evidence should take into account both the functional effect in the assay and the mechanism of disease. Finally, in terms of our general recommendations, we believe that the entity performing the functional assay shouldn't govern whether the criteria is satisfied or what strength. Rather, the determination should be made based on validation of the assay, including use of appropriate laboratory controls as well as clinical validation controls. So this was really meant to address the idea that CLIA laboratory tests carry the most strength of evidence, whereas really we feel that it should be demonstrated validation that leads you to apply the criteria and the level of strength. So next I'll walk through the 4 steps that we developed to curate functional evidence and decide whether these codes should be applied and at what strength. So first, defining the disease mechanism, then evaluating the general class of assay used in the field, then evaluating the validity of specific instances of an assay, and then finally applying the evidence to individual variant interpretation. So for defining the disease mechanism, in order for a functional assay to be useful, you must understand the underlying gene disease mechanism. And we suggest that this be described in a structured narrative with these 5 points. First, the gene name, and the associated disease in Mondo terms, the mode of inheritance, so autosomal dominant, recessive, mitochondrial, X-linked, etc. The molecular mechanism of disease pathogenesis in controlled terms like loss of function, gain of function, dominant, negative, as I talked about before, and then biological pathways that are involved. And so, for example, for CDH1, your disease could be hereditary gastric cancer or breast lobular carcinoma. It's an autosomal dominant inheritance. Loss of function is the main mechanism of disease pathogenesis. And some of the biological pathways that might be most relevant to disease pathogenesis might be cell-cell adhesion or these calcium-dependent adhesion mediated by cadherin. So when you then go to look at these assays, this is what you're considering. And this really only needs to be done once per gene disease pair. So you could potentially do this in the future in a kind of pre-curation space, and ideally it would get loaded into the VCI such that when you get to the point of curating your variants, this step is already done. Next is evaluating the general classes of assay used in the field. So does the general class of assay model the pathogenesis or disease mechanism? Things you want to consider are the model system, cell lines, in vivo or in vitro work, and the strengths and weaknesses of each of those systems. What are the experimental methods that are being used? What's the functional outcome being evaluated? Does that reflect the disease pathogenesis, such as loss of function, gain of function, or the output of the functional pathway that's involved? Ultimately, the goal is to identify what types of assays are appropriate if they're sufficiently validated to be used in clinical variant interpretation. Other things that groups might want to consider is if they don't see any available functional assays that appropriately model disease pathogenesis, for example, are there any that they could imagine that if we had it available, this would be really useful? And delineating that would be important in your curation as well. To be able to endorse an assay that could be used in the future, though you would want to cap that level of strength it's supporting until that assay could be formally evaluated in the future. And then are there any limitations on the assay with respect to applying the evidence in favor of a benign interpretation? So assessing whether the given assay can evaluate all the relevant functions of the protein or provide a readout that's reflective of the disease mechanism as a whole, because if they're only looking at specific subfunctions of the protein that are involved in disease pathogenesis and it's not capturing everything, then you may only be able to assert that it's not abnormal for this one function, but you don't know if it's benign for the other functions. So then if you are looking at the specific instance of the assay, so many different labs might generate functional evidence using the same general class of assay, but differences in specific methods used and the level of validation provided by each group leads us to evaluate each individual assay iteration before it can be applied in clinical variant interpretation. So our considerations include our basic controls included. So having a normal, negative, or wild type control, an abnormal, positive, or null control, and multiple replicates, biological or technical replicates, and specifying how many there are. And where variant controls used, so known pathogenic and known benign variants. And those are important because they help establish the dynamic range of the assay and are really important for clinical interpretation. If you only have one variant and one wild type control, you don't know what your range of pathogenic variants would look like. So this is an example of an assay that shows a number of non-pathogenic or known benign controls and a number of known pathogenic controls that allowed them to establish thresholds above which they know these variants to be functionally normal and functionally abnormal, to be able to classify these variants of uncertain significance in the middle here. If these parameters are not met, then there are still some ways to give evidence for functional assays. If it's broadly accepted historically, if it's been previously validated, such as the lab has previously published on the assay showing all these validation controls, but then later publishes, again, looking at another couple of variants, but doesn't show all the same validation as before, or if it's provided as a kit with defined performance metrics, but the controls or replicates are not documented for the specific instance of this assay. So maybe a clinical lab tests enzyme activity, or you buy an FDA-approved kit, something like that, and the group that is using the kit has not demonstrated all of the validation that went into the making of that kit. Then you can still find some evidence at the level of supporting. But if it doesn't meet any of these criteria, then you don't want to be applying the PS3 or BS3 criteria. If these conditions are met and there's at least 8 total pathogenic and benign variant controls used, then we found that you can proceed to a more nuanced approach for how to apply the evidence to individual variant interpretation. And I'll get to how we came up with 8 in just a moment. So if there's enough statistical analyses to estimate or calculate an odds of pathogenicity, or if there's not sufficient statistical analyses to estimate or calculate the odds of pathogenicity, then you can first look at how many total benign or pathogenic variant controls were used. If there are at least 8, then we found that to be at the level of supporting evidence, and if there's at least 12, then that would give you at least moderate evidence. So to determine the minimum number of validation controls that were needed for a functional assay to provide a supporting or moderate level of evidence in the absence of rigorous statistical analyses, we estimated that the odds of pathogenicity could be obtained from evaluating various numbers of previously classified controls. So we assumed that the assay readout might also be indeterminate for about 20% of variants, and the remaining known pathogenic controls would have a functionally normal readout, functionally abnormal readout, and the remaining known benign would have a functionally normal readout. So here you see a snippet of the table that we used to make these estimates. So if you had 6 total classified variants to start with, and 3 of them were pathogenic and 3 of them were benign, then your prior probability of 0.5 would give you an ultimate odds of pathogenicity of 2 and an odds that it's benign of 0.5. And then looking at this example where you have 31 total controls, 13 pathogenic and 18 benign, your prior probability would have been 13 out of your total 31, gives you the proportion of your pathogenic out of your total variant controls, and that's 0.4. If you say that your assay result is all of these functionally abnormal, no, it's this one, yeah. The functionally abnormal results are below your threshold, so they were called damaging or abnormal, then all 13 would go here. This one doesn't have any indeterminate necessarily for its controls, and the functionally normal, all 18 benign were normal results. This is your posterior probability, which here you just add an additional 1 unclassified variant to help avoid having any infinity or 0 results when you get to your odds of pathogenicity. And you can calculate your odds of pathogenicity using your posterior probability and your prior probability from before, and get an odds of pathogenicity of 18, which is equivalent to PS3 moderate, and odds of pathogenicity for a benign result, 0.077, which would be a BS3 moderate. I know there are no moderate benign codes in the combining rules, and so that would be equivalent to 2 supporting pieces of evidence. If you do have enough statistical analyses provided by the paper to estimate or calculate an odds of pathogenicity, then you can correlate the strength of evidence to the calculated odds of pathogenicity using this table. And the maximum you could get is a PS3 very strong, and a maximum benign would be strong evidence.
This is Deb. Can I ask a question? So everything that you're talking about in the last 2 slides, this is combined over all of the functional
papers that you and assays you've come across? It's not like this all has to come from one paper?
So this typically is looking at one paper, one specific instance of the assay, so one laboratory conducting it, because often you might find that they are run in very different ways between labs.
Right, yeah.
And so, yeah, we are really looking at individual laboratories.
I mean, I'm just thinking in terms of, I don't want to like get this too off track, but like in terms of VHL functional data, I mean, there's very few papers that test to like, 8 to 12 variants and up.
Right.
So I was thinking that it was the slide before this one, I was thinking was like a cumulative across all of the assays that are all like similar assays,
Right.
HIF1 alpha degradation assays from different laboratories and papers. But okay.
Yeah, no, this particular figure came from one paper, one type of assay. They tested a bunch of different variants. But your point is an important one, and one that I am going to speak to a little bit at the very end.
Great, thanks.
So this is an example of a paper where the group was able to use receiver operator characteristics to determine their sensitivity and specificity of their assay and equate the numerical assay outcome, which was the percent of wild type repair with the probability of pathogenicity, and then they converted that to an odds of pathogenicity. So each individual point was then able to have its own odds of pathogenicity. So it is possible. This is obviously more possible now with some of the techniques we have and as we move towards more high throughput functional assays or multiplex assays. I think we'll be able to see more of this, but most assays where you're able to calculate a sensitivity and specificity, you can start moving towards the actual calculation of an odds of pathogenicity. And if the same variant is analyzed by multiple different assays or the same assay but by different groups and the results are consistent, then you want to apply the functional code at the level of strength of the most well-validated assay. If the results are in conflict, one shows that the variant is functionally normal and another shows functionally abnormal or an intermediate result, then you want to go with the result that more closely reflects from the assay that more closely reflects the disease mechanism and is more well validated and can override the conflicting result, applying that at the level of strength indicated by its validation parameters. If at essentially the same level of validation between the two assays, then you don't want to apply any of the evidence for that variant. So for variant controls, it's true that historically most functional assays cited in variant curations have really analyzed very few variants, but there are new multiplex assays that can analyze thousands of variants in one experiment. And so I think that we'll start to see a trend towards more of these assays that will be eligible for the higher strength of evidence as we move forward. There are some issues for certain genes where there are very few benign and pathogenic controls available at all in ClinVar. And one of the things you might be able to do in developing a functional assay, or if you've got many different variants that have been assessed, is looking to see in NOMAD or in other databases, just based on the frequency, whether it would qualify for a benign or likely benign based on that population frequency. And then for those that have very few pathogenic controls in ClinVar, you may be able to look at the results for variants that meet likely pathogenic or pathogenic without functional studies, but maybe haven't been deposited in ClinVar yet. Ultimately, our goal is to be very rigorous with the functional data that we do accept in clinical variant interpretation because it's so important that we get it right. We don't want people to make decisions based off of incomplete data or data that hasn't been conducted to a high standard. So we really want to be conservative in our application of the PS3 and BS3 criteria. And so I know that that's going to be a bit of an issue for now as we move towards more of these assays where you're going to have the ability to calculate sensitivity, specificity, and odds of pathogenicity. But that is kind of what we're looking to do, is to really hold laboratories to a high standard to be able to use that data in clinical variant interpretation. So both the results that I presented at the very beginning and the recommendations are currently available on preprint servers, the first on medRxiv and the second on Bioarchive. And Shannon from the earlier group will be presenting a poster at ASHG, and I will be talking about this at ASHG in an oral presentation. If anyone's there and wants to learn a little bit more or meet and talk about it at all, I'll be at that meeting in October. And if you want to just end here with the overall flowchart of how we recommend approaching the variant curation process, here is all of it together. So I'm happy to take questions.
Great, that was a fantastic overview, Sarah. Thank you so much.
Thanks, Jen.
I have a question. How does this apply to localization studies where you're determining whether the protein localizes properly or not?
Yeah, that would be one example of a general class of assay, and so you would want to ask whether that supports your disease pathogenesis mechanism, and again, you would want to look at the same controls, replicates, et cetera, for those kinds of assays.
Okay, thank you.
Sarah, I know you had said that this scheme should really apply to each instance of the assay, but if you have, excuse me, a lab, like a well-known research lab that's been working on a particular disorder for a long time, and among the results that they've published, they're not really reporting pathogenic and benign variant controls as such, but in among the variants that they have reported, you know, like these are kind of now well-known pathogenic or benign variants. Is it possible, or do you think it would be appropriate to just put all of that evidence together? So let's say there are like 10 papers, and among those 10 papers, you can find your 8 variant controls, but yet they're not actually for each instance in each paper publishing a pathogenic and a benign control. Do you understand what I'm saying? Is that an appropriate way to do this, or is that something that would need to be discussed before it's decided if that's okay?
I think that that may be the reality for now, and that, yeah, the important thing, though, is to make sure that those that are the well-known benign and pathogenics that you're using to count as controls are controls based on other evidence besides functional evidence. Because I know that often that's how maybe the first couple become known as pathogenic, is maybe someone tested in an assay and showed that it has a negative impact on the protein's function. And you have to look at the clinical evidence too.
Sure. So along those lines, go ahead.
Hi, this is Jordan, and do you assign different weights for in vitro versus in vivo assays?
So no, it would not be a different weight based simply on whether it's in vivo versus in vitro. You want to look at how well validated each of those assays is.
Okay, thank you.
Sarah, this is Jenny again. So I have another question. Let's say that for one research group or one paper or whatever, you feel like it's a well-validated assay, and then another publication comes along and they match some of the results. So it's the same variant and they're finding the same thing. It's falling in their pathogenic range or whatever. Is it possible then to use that evidence because it's simply matching what another group found, even if it's not a known pathogenic variant. Let's say it's still a VUS, you don't have enough information about it, but two separate groups using the same assay are finding the same result. I guess that's not really validating the assay as such, but kind of validating the process, I guess. So maybe not. I'm just kind of curious because I've seen that for some of the GAA data, that there are maybe 3 separate papers and actually different labs, different groups where they're all finding deficiency, but it's not clearly a pathogenic variant. We just don't have enough data.
Right.
So I don't know if that's something you've thought about.
Yeah, that is something that we thought about at the beginning, and then kind of moved more towards making sure that the specific instance that you're going to count is the one that's the most well validated, that you can't validate the assay just based on being consistent with results that have been done by other labs.
Okay.
Unfortunately.
Yeah, it's good to know.
This is Diane from the PA. Can you hear me okay?
Yes.
Yes, we can.
I'm just wondering, for like pre-existing groups, are there recommendations of how to now incorporate these into previous variant submissions?
Right. So that's a great question, and one that we were discussing this morning, actually. So we will probably work with the SBI and those existing groups to go back and review and maybe test out some of these rules again to make sure that we're applying the best evidence that we have for these variants. So I think that for now, groups should continue as they have been doing, but moving forward, we'll probably end up reaching out to the existing DCEPs to see how we might need to modify some of those criteria. Does that answer your question?
Yes, sorry, I put myself on mute again. Thanks.
And this is Jenny. So, I mean, it's an interesting question. I mean, one of my favorite genes has been studied for decades, and I think, early in the process, in the 80s, 90s, they weren't necessarily doing these sort of huge, big, many sample assays. It might just be a few patients, and a lot of the variants they're looking at are ones that, are VUSs. That's why they're looking at them in the first place. They don't really bother looking at the ones that have already been found in 20 patients. So the information just isn't really there. So moving forward, I mean, it really seems like for some of these diseases, you would really want someone to develop like a fully validated assay, right? Although this is Pompe disease, and we've been looking at the expression of these variants in a cultured cell and looking at the activity of the enzyme, there really isn't, the type of evidence that you're showing here. We'll certainly try and get some of that together, but that would be the goal moving forward, is that somebody's going to have to do this if we, otherwise we just aren't going to be able to count that. And I wonder, right, as the field, how is that going to work moving forward, for funding, and who's going to do these types of things?
Yeah,
that's just a comment and a thought.
Yeah. It's a great question, and I think that one of the goals of these recommendations, too, is to help partner with more bench and basic scientists so that they also know what it is that we need to be able to count this kind of evidence in clinical variant interpretation, because they may not be thinking about that when developing an assay. Because you're right, you might see a variant in a patient and want to go run a test in your lab and just see what the result is, but unless you're comparing it to a bunch of knowns, then you don't really know where it falls within those that you already know are pathogenic or benign. Right. And the benefit that you have of having these existing assays that you've tested VUS in before is that you already have an assay that's working. You maybe just need to test it a couple more times with additional variants that are of known effect to be able to determine a good sensitivity and specificity.
Sounds like there might be a need for, I don't know, some kind of biotech company or somebody to take this on, as, I don't know, maybe there are companies doing this type of thing already. I don't know. Any other questions?
So I think, this is Deb. I think for groups, so for instance with VHL, we're pretty far into our functional data review, and we didn't have any of this information until I saw the consortia call on Friday. So I'm just wondering, I mean, I think it would be good for anything that's going to have a major impact on our biocuration work if the biocuration working group has some intro into it. I mean, I know this has probably been discussed quite often on the SVI call, which I haven't been on. But then again, I don't know how many biocurators are also on the SVI call. So, yeah, I feel like this is something that's kind of dropped a bomb in the middle of the functional work that we're doing. And it would have been good to have just been aware of this, or kind of looped into it a little earlier. I feel like in terms of questions for this, I'm not really going to even know the questions until I have a chance to try to see whether our data conforms to it or can kind of get worked into it. And then I think it'd be good to meet again to go over some questions for it.
Yeah, and that's a good point, Deb. Sort of moving forward, just making sure that everyone is aware of things that are coming out. I mean, I guess one thing with that is, it takes up, obviously, Sarah can talk to this better than I can, but this would have taken quite some time to put together. So, what to present. I guess even saying that this is coming down the pike, and just be aware that these recommendations will come out, but we might not want to give information that's not yet finalized. So,
yeah,
but I do see what you're saying. Yeah, but at least being aware that something is coming, and like you say, not being suddenly dropping the bomb like, oh my goodness, I didn't even know that this was coming out. So yeah, we can definitely think about that moving forward for this group, just in terms of announcements. Yeah.
But I mean, it might be good to say, get a sense of what are the groups that are currently kind of in progress with functional data, and then to have them, look at this, try and see how they can adapt what they've done so far to it, and then be just with those to then go back to say, now what are the questions now that you've taken your real-time data and from all this kind of different groups. Like I'm thinking, are there particular considerations we need for cancer data, for, tumor assays and things like that in there. So.
Right. I appreciate that feedback. And yeah, as Jenny said, one of my hesitations about maybe getting this out earlier was that it wasn't finalized or submitted yet. And I know that even right before we went to submit, we made a couple of changes in terms of giving a number, at least for groups to think about in terms of number variant controls. But I would love to connect with you and any other groups that are also in progress, because that kind of information and how we can help you and how your information can help us better refine these guidelines is going to be really important. And as we get ready to make revisions on this manuscript, that information is going to be really key so that it's feasible also for groups.
This is Diane again from the PAH work group. I also feel like along those similar lines, just the comparative analysis, like the PAH group obviously didn't do it to this extent, but had taken into consideration some of the topics that were put up in that first chart, although we maybe didn't expressly suggest them in our publication. So I'm wondering if there was ever a discussion with the different groups to assess, like, I don't know, some of those additional topics. I don't know if that makes sense.
Yeah, I don't know how much. I think that Shannon or Donna did reach out to a couple of groups potentially to get some additional information, but mostly they went from what was available in the publications originally. But I mean, we'd love to get any additional feedback of things that, especially that were considered, but then not ultimately used. It's a lot